Thank you very much for the invitation. It's a pleasure. Thank you all for being here. And thank you to the ATSE staff who have done so much to make this happen. I particularly want to apologize to the long-suffering Dominic and Sue, who have been so patient with me as we were setting up this interaction. 11 years ago, together with my colleague Michael Holmes, we at Sangamo here in Berkeley, California, invented the term genome editing. The dream at the time was we would have the experience of sitting in front of a living organism and having a keyboard with nothing but the four bases of DNA and be able to type genetic code inside the nucleus of the organism. Little could we imagine today, 2016, that to what extent this has become reality. And what I'll do over the next 25 minutes or so is walk you through what we know about the general principles of gene editing, and then give you some specific examples with a particular focus on things relevant to agriculture and with some forays into therapeutics, but only where I think folks who work on engineering plants or animals could learn something from the experience. I'd like to start with a plant perspective. Joseph Petolino, who was the project leader of our partnership with Dow AgriSciences, guest taught in my class. I'm also a professor at UC Berkeley. And he told my students this remarkable story that I think is really relevant to the notion of using gene editing. And it has to do with dehiscence in rice. Obviously, naturally occurring plants, they shed their mature seed. But rice, of course, is grown in a way where should rice just shatter the seed into the water, then it will be difficult to harvest. There are multiple loci in the rice genome that are responsible for shattering, and it's clear that lack of shattering or indehiscence is an acquired trait that emerged through domestication. The truly striking thing is when the loci were mapped, the strongest allele turned out to be a point mutation. If you create one point mutation in a transcription factor in the rice genome, then you have this domesticated creature, this organism that doesn't drop its seed into the water. And human selection apparently favored this mutation. So if you want to have an organism such as a crop, which has an agronomically useful trait, the traditional approach, since, for example, rice was domesticated 10,000 years ago in Southeast China, has been to find naturally occurring genetic variation and then somehow breed it into your crop and breed away things that you don't want. So the age of genome editing has fundamentally changed this paradigm. If your sequencing effort has identified an allelic variant that creates a novel trait of interest, then you can produce it in your organism of interest in a single step without essentially any breeding. So, and as an example that I will walk you through in a second, I will tell you how, together with scientists at the Roslin in Edinburgh, in particular Bruce Whitelaw and Simon Lillico, we did this act of moving a tiny piece of genetic information from one creature, the warthog, to another creature, which is the pig, to create an agronomically useful trait. And I will then explain to you how this can be relevant for plant ag as well. This is only one example, but I just think that it's time for us all as a community of people who work with organisms in lab or in the field or in the commercial production facilities. We have to approach their genetics fundamentally differently. Because for the first time, I think, in the history of biology, we can introgress, so move genetic information from one taxonomic entity to another, not via mating and backcrossing or any of those old-fashioned things, but in a single step via editing. And so the other thing that I'd like to point out is we actually did this on humans. We've done some introgression of genetic variation into human beings, and I'll talk about that in a second. And of course, this is just the start of what editing ultimately will achieve. So let me walk you through some of the things that we and others have done with editing. You know, we started on this 16 years ago, and we knew that we were faced with some severe problems. And so bear with me while I spend maybe 5 or 6 minutes on some basic biology, and I suspect that many of you are familiar with this, but I promise to try to provide at least some novel perspective on this. So the first problem we have in trying to change DNA in a targeted fashion is the problem of just sheer genome size. Before you is just 12 base pairs of DNA, very, very familiar to you. Of course, the human genome, for example, is 3.3 times 10 to the ninth. And then on the next slide, you see about 0.3% of the human genome in a form released from the cell. My favorite analogy in terms of how big DNA is comes from an American radio show called Radiolab. And the folks who produce it have pointed out that if you take the human genome and if you read one letter a second, it will take you a century to read the whole human genome. It's huge. And so what we're trying to do is we're trying to specify inside the nucleus of a living cell essentially a single position in that enormous amount of information. The other problem is the problem of genome defense. Bacteria and yeast, being microbes, welcome new DNA and accept it with great glee. That's absolutely not true for pretty much any other taxonomic group on Earth. So, for example, if you take normal mutant human cells and put in a wild-type piece of DNA, then 1 in a million of those cells will acquire the corrected allelic form at the endogenous locus. So that's a bit of a problem, this kind of efficiency, 1 in a million. So the solution to both recognition of a target locus and the ability to change it is, of course, genome editing. The first thing I need to explain, and I will explain how we recognize loci and how we create the editing outcome through double-strand breaks. So one thing I would really encourage, and I know that there are many folks who lead major efforts in science policy or the funding of science, I really want to bow before the academic scientists whose work led the way to the development of gene editing. None of them wanted to create a cure for genetic disease or novel ways to genetically engineer crops. They exclusively were basic scientists. So the first one is Aaron Klug. He discovered the Zincfinger. The second is Carl Pabo. He solved its structure and proposed a way to design it. Maria Jason was the first one to show that you can do editing via targeted break. And the culmination was work by Dana Carroll, who invented Zincfinger nucleases as tools for genome editing. And then the rest is multiple exclamation points. So the solution to the problem of the genome being resistant to change is you need to activate the target for genetic engineering by cutting it inside the nucleus of a living cell. When you cut the DNA, you evoke an ancient and powerful biological process known as double-strand break repair. There are many ways in which organisms repair damage to their DNA. And double-strand break repair is particularly powerful because double-strand breaks are highly genotoxic. If you have a chromosome and you have a break, if you don't repair the break, then whichever fragment does not have the centromere will get lost and the other fragment will lack a telomere. So double-strand breaks have to be fixed. I'd like to show you one piece of data, but first let me show you a schematic. In brief, there are two fundamental ways in which breaks come together. The ends can just come back together again. That's called end joining or non-homologous end joining. Alternatively, the break can reach out to an extra chromosomal piece of DNA, and that can go in. And that's called homology-directed repair. I'd like to show you a really beautiful piece of data that I always use to introduce gene editing because it really reminds us of how powerful Mother Nature is and how extraordinary double-strand break repair is in particular. So this is a yeast DNA. So here yeast cells, and they have been separated, their chromosomes on a pulsed field gel. So let me be clear, these are not 1 kb. These are 1 million base pairs, 800 kb, 600 kb. These are huge. And so if you irradiate the yeast with X-rays, and you see the increase in dose from left to right, not surprisingly, the X-rays create double-strand breaks, the chromosomes get sheared, and you see this rather ugly-looking smear. So obviously you will say to me, the yeast will die. So if you just turn off the source of radiation and give the yeast a little bit of time, then over the next 2 hours, this little organism puts its genome back together again. This is the biological process that we evoke when we do gene editing, except, of course, we don't carpet bomb the genome with X-rays. We create a single double-strand break, but we still activate this pathway. Jim Haber wrote a haiku in which he explained why that exists. He goes, chromosome broken awaits sounds of strand pairing, preserving life's thread. That's the biological rationale for the pathway. How do you create a cut at a single locus inside the genome of a living cell? There are 4 solutions, and they all work. And the single most important message, folks, that you can take away from my talk, other than the fact that genome editing works really well, is that the only thing you need to get genome editing to work is a really good nuclease. And there are now 4 separate ways to do that. So do not spend any time nuclease engineering. You can get them off the shelf. The best studied class, the ones in the clinic, the ones with the longest track record, are the Zincfinger nucleases. Meganucleases were in fact deployed first for genome editing, but they turned out to be really difficult to engineer. We invented tau effector nucleases for use at endogenous loci. It's really noteworthy that the initial work that identified the transcription activator-like effector motif came from a plant pathogen, Xanthomonas. And of course, if you haven't heard of the molecule that's on the lower right, you must have been in fair amount of seclusion from the world, because this, of course, is CRISPR-Cas9. And I'll have more to say about this in a second. I'm going to spend a little bit of time walking you through how you do gene editing with Zincfingers, but I suspect most of you want to hear about CRISPR-Cas9. Here's the good news. 100% of what I say about Zincfingers and what we learned with them applies to CRISPR-Cas9. The reason that Cas9 took off so fast is because they had 8 years of precedent of how to do genetic engineering with Zincfingers. So I'll skip how you engineer Zincfingers because, like I said, if you want to edit a locus, you just order this thing and in it comes in through the mail. It basically has a DNA binding domain, in fact, 2. They bind to the same locus in a precise orientation and spacing and then create a double-strand break. And that's where it gets, that's where the interesting things begin. So this thing binds to DNA, and if you want to mentally replace what you see before you with CRISPR-Cas9, that's absolutely fine. It binds to DNA and then it cuts. And that double-strand break must be repaired, as I mentioned to you. And that repair... Did my slides... Could I have the next slide, please? Aha, thank you. And that repair, though that cutting will happen over and over again until we get a biological outcome. And the biological outcome is one of 2. We either get a gene knockout as a result of non-homologous end joining, or we get gene correction or addition if we've provided an external template, the information from which would go into the chromosome. So very briefly, I know most of you don't work on therapeutics. This is the bubble boy. This is David Philip Vetter. He was born with severe combined immune deficiency, so he had to live in a bubble and could only go outside in a special spacesuit engineered for him by NASA. We set out to try to create a gene editing-based approach to the treatment of this disease. And unfortunately, the gene has many different mutations that affect it. So one key fact that you should be aware of is this beautiful piece of data from Maria Jason, which is when you cut a locus, then the wave of genetic correction is actually relatively short. In human cells, this window within which gene correction occurs from the break is only about 200 base pairs. So this means that we had to engineer a nuclease that would precisely bind to the position of the mutation. Folks, and this is true for all of your applications. You could be working with wheat or you could be working with hamsters. It doesn't matter. You have to cut as close as possible to the position where the editing is to happen. And we did that. So we made Zincfingers that cut precisely at the position of the mutation. And I'll show you just a couple of gels. It's useful to look at primary data because it gives you a sense of the assays that one uses. So here we're going to cut the chromosome in living cells with a zinc finger and then provide an extra chromosomal template which has a single point mutation which is going to go into the chromosome. And if it goes into the chromosome, then the chromosome will be sensitive to a restriction enzyme. And we did this experiment and we're completely floored to see 20% of the chromosomes acquire the novel mutation that we have specified. This was a PCR-based assay. We don't trust PCR all that much. So we grew the cells out. We did a southern blot, which is shown on the next slide, and the results didn't change. We were completely ecstatic because this had no precedent in the history of science, such efficiency. So we then did single-cell cloning, and this is my other very important point to you. We were able to achieve very high efficiency of both homozygous and heterozygous corrections at the endogenous locus. So we've since taken this to the clinic, but what I'd like to do next is walk you through this pig and warthog example of doing gene correction for agronomic species. So Bruce Whitelaw at the Roslin came to me, and we've collaborated with him and Ian Wilmot for a while, came to me with a request. They were worried about the African swine fever virus. It's endemic in sub-Saharan Africa, highly pathogenic in domestic pigs. So my understanding is that the approach to eradication is to just cull the herd, which is, quite severe, putting it very mildly. The warthog, however, is resilient. The warthog does not really succumb to the infection. So RelA was identified as a potential mediator of resilience. And when they sequenced the warthog allele of RelA and the pig one, and they only saw 3 point mutations, well, that's amenable to gene editing. So we made Zincfingers that cut the pig RelA, and then designed a corrective template that would transfer the point mutations into the pig genome. And then went ahead and did injections of the Zincfingers and the repair template into one-cell pig embryos, which were then implanted, and the resulting piglets were genotyped. And the results are published, and I'll just tell you what the results are. To everyone's complete glee, piglets were born that were beautifully heterozygous for one allele that was pig and one allele that was now warthog. Piglets were born that were essentially homozygous for the warthog allele on both homologous chromosomes, except for 1 point mutation that did not convert to the warthog form. But that was a non-issue because there was a piglet in the litter who was homozygous for the warthog haplotype. So in a single round of gene editing after injecting an embryo with nuclease and a repair template, 3 point mutations were essentially transferred from the warthog genome to the pig genome. And these aren't just mutations. These are supposed to be alleles that should confer resistance to the African swine fever virus onto this piglet. The animals are being grown for resistance testing. So this is an overwhelming slide and yet is out of date. It's a year and a half old. These are organisms that have been genome edited. The list is doubtless larger. What I just described to you in the pig is feasible in all of these critters. You just have to have delivery working. So typically in vertebrates, there's just embryo injection. And for plants, a broad range of delivery technologies are used. But the really stunning thing about this slide, folks, is if you look, we have Drosophila and Arabidopsis, the silkworm and wheat, salmon and cabbage. This is the beautiful thing about the unifying principles of gene editing, is that you need a nuclease to cut the chromosome, and you need a repair template to repair the break or not provide a repair template, and Mother Nature does everything else. It's really remarkable. So I'm looking at the clock, and it's looking to me that I'm looking to my AV folks. Do I have about another 5 or 10 minutes? Give me a sense of, I have a whopping 10. I'll slow down then. Excellent. All right. So I told you about targeted correction in the pig. Let me show you 3 slides of data of gene editing in crop traits. You will hear from the next speaker about the broad range of ways in which GE crops are being deployed. Let me just give you 3 examples of how gene editing has been deployed. It's been largely focused on gene correction or addition. All of these are published, and I'm very happy to report that subsequent work has really validated the fundamental feasibility of this. You basically cut the chromosome with Zincfingers. You deliver a repair template that in this case specifies an herbicide resistance marker. Then you apply selection, and you have this beautiful transfer of exogenous genetic information to the chromosome. This was in tobacco, I believe. Then we went ahead and did this experiment in corn. And I will never forget when we got these data. We were able to show in partnership with Dow Agro that we can drive the targeted addition of an herbicide resistance marker into an endogenous locus in the maize genome in a single step biallelically. So single step biallelic modification of an endogenous locus. And this, I should also point out, this created a trait stack because the locus we were editing encodes an enzyme in phytid biosynthesis. So this was simultaneously herbicide resistance and low phytate. And last but not least, this also enables, in a very nice way, trait stacking. Again, this is published. The idea is that the transgene that goes in contains the recognition site for an additional nuclease. So once you have an initial event with a transgene at a locus, you can then add more things to that transgene as if this were a USB port. So, and I'll skip through this in the interest of time because this is published, simply to point out that this works really, really well. So the bottom line, folks, the one objective limitation with respect to gene editing in plants is delivery. With embryos of pigs, you inject. With human cells, you deliver in a dish. With maize, you do embryogenic callus for HI2, but that, as you know, doesn't exist for other germplasms. Immature embryo bombardment has worked really well. You can do delivery to hypocotyls. People have tried agro. People have done protein delivery. But bottom line is the one objective barrier to end, well, not so much of a barrier, something that you have to cross as you go from your trait of interest to the edited plant is how are you going to deliver the nuclease and the repair template. And my only guidance to you in this regard is don't reinvent the wheel. There's been an enormous amount of work in this regard. So just the literature should be your guide. Let me close on a really remarkable recent set of data, which I think have major implications for organism engineering, which is how efficient gene knockouts are. So the next slide should show our a clinical pipeline. Again, I accept the fact that most of you don't work on human therapeutics. My central message to you is that gene editing has been in the clinic for, I think, 6 or 7 years now. We've treated over 80 subjects. We have multiple open investigational new drug applications and clinical trials, both ex vivo and in vivo gene editing. In the interest of time, I will go through this really quickly. I just want to give you some numbers to give you a sense of how powerful this is. I'm going to focus right now on gene disruption, which is the pathway that relies on non-homologous end joining. I told you about introgression in the context of agronomic efforts. The gentleman on the left is Timothy Brown. He was cured of HIV because he received a bone marrow transplant from somebody who was homozygous for a naturally occurring protective allele of a gene called CCR5. So the gentleman on the right is Matt Sharp, one of the 80 subjects on our trial. And what we do is we basically introgress by gene editing this HIV-resistant allele into Matt Sharp's genome. And it's on chromosome 3. And the way you do this is you harvest some cells, you gene edit them, and you put them back in, and they create small insertions and deletions that then drive the knockout. I'll just show you one piece of data, which is really stunning. What it shows is we take a human with HIV, take out their T cells, gene edit them to knock out CCR5, put the cells back in, and then the subject can stop taking antiretrovirals, and that's the black line. And the virus load just goes down to below infectious level in the absence of any retrovirals. So we've just genetically re-engineered this person's immune system. So I get a lot of questions about safety and off-target effects. I really encourage you to look at the literature for gene editing in human cells, because I'll just go through this slide as quickly as my clicking will allow, simply to say that there's a really comprehensive set of specificity assays that have been developed. And if you have an interest in understanding what gene editing does in your organism, then you can basically dive into that literature, and you will learn what kinds of assays could be useful to you. All right. So one last slide about efficiency, one last slide about plants, and then we're done. So we wanted to gene edit T cells. And what you're about to see is it's completely unprecedented, which is efficient single-step knockout of 2 genes at the same time. The logic here, as far as editing human T cells to treat cancer, is you want to weaponize the T cell with a chimeric antigen receptor. But the problem is that the T cell can be recognized by the organism as its own T cell receptor could be cause it to be rerouted to a different tissue. So we set out to do gene knockout. The goal is to knock out the TCR and the HLA to denude immunologically the T cell. How well does it work? Well, here's the 2 important pieces of data. We take a clinical scale dose of T cells, deliver the Zincfinger nuclease, and we get 92% with no selection. 93% single-step knockout at endogenous loci. So the message to you is, with the right delivery and with well-optimized nucleases, you can get completely astonishing efficiencies of targeted genetic engineering. The key result here, which I think is just really sort of take your breath away, is you can deliver nucleases for locus 1 and locus 2 at the same time, and you will get 82% single-step 2-locus full knockout. So I think that this multiplexing of editing, where you go after multiple loci at once, is really the way of the future, because many traits are polygenic. And my message to you is, you don't have to make them iteratively, trait 1, trait 2, trait 3. You can make traits at the same time. One really striking result in this regard, and I think this is my penultimate slide, is this really impressive work from Dow Agro. Selectus has done this as well, as have others. How do you do plant gene knockout? So what you do is you take a plant transgenic for a nuclease, which will cut at the target, knock it out, and then you segregate the nuclease from the knockout target allele. So the null segregant has none of the transgene and just has an indel, a small insertion or deletion, at your gene target. And so I can't speak for Australia, but I know that in the United States Department of Agriculture, APHIS, has ruled that maize made using this approach with a small indel at an endogenous locus, or potatoes and soybeans made by Selectus, all they have are small indels. They are not regulated articles and under Code of Federal Regulation, which I think, first of all, is entirely reasonable. They contain no foreign genetic material. All they contain is a small indel at the endogenous locus. But also, it just really ushers in the new era of genetically engineered crops. All right, my last slide. I told you that engineered nucleases such as Zincfingers, TAL effectors, or CRISPR-Cas9 induce a double-strand break at endogenous loci in cells, and that evokes 2 pathways, non-homologous end joining, which can be used to drive gene knockout with pretty remarkable efficiency, including multiple gene knockout at the same time. If you co-deliver an additional template, you can do gene correction. I gave you the example of the warthog haplotype being transferred into the pig. Or you can do targeted integration. And I showed you several examples from the plant world. Everything that we learned about Zincfinger editing has been successfully ported into the realm of Cas9. That's, as I said, why the field has moved so quickly. I have a wealth of people to thank. I think most importantly, I'd like to acknowledge my colleagues at Sangamo, Ed Rebar, Jeff Miller, and Lei Zhang for making the Zincfingers. Mike Holmes for being my partner in action, my fellow musketeer, and also Dale Ando and Jeff Nickel. And again, to acknowledge wholeheartedly the academic scientists on whose shoulders all of this work lies. Thank you very much.